Hello, everybody. It's great to be here one more time today. My name is Gary Fowler. I'm the CEO, President, Founder of GSD, Get You Done Venture Studios and Family Office. I'm a 17-time serial entrepreneur with several unicorns under the belt. I was on the original management team at Click Software, which was sold to Salesforce for $1.35 billion, and also Eva.ai, the first generative AI company that I founded in 2016 with Dr. David Yang. We believe at GSD that intellectual capacity is evenly spread, but opportunities are not. Today, I'd like to welcome you to my show, GSD Presents Top Global Startups, where I bring you the visionaries building the future one breakthrough at a time. Today's episode sets at the intersection of biology, artificial intelligence, and industrial state innovation, a place where science fiction is rapidly becoming reality. My guest today is Daniel Delubac, CEO of IORGANBIO, a company redefining what it means to model, repair, and even recreate human biology. Daniel is not new to breakthrough innovation. His work has contributed to some of the most transformative advances in modern healthcare, from non-invasive prenatal testing at Council to liquid biopsy at Guardant Health, early cancer detection at Freenome, and patient-derived tumors at his last at Exelis. He's been at the frontier of AI-driven chemistry at Chemify, and now at his new companies tackling one of the more ambitious challenges of our time. How do we manufacture human bioequivalent cells at scale, cells that can model disease, accelerate drug discovery, and ultimately restore human health? This isn't just about biotech. This is about building the infrastructure layer for the future of medicine. With that, I'd like to welcome Daniel to the show. Hi, Daniel.
Hi, Gary. Thanks for having me.
Daniel, you've been part of multiple categories of companies redefining. What threads connect all of your work together?
Yeah, great question. I think, generally speaking, my career has been pursuing opportunities, if you will. And the first one that has driven all the future one was at Council. An exceptional individual, his name is Balaji Srinivasan, a lot of people will know him, gave a presentation at Carnegie Mellon right after my PhD when I was thinking about what I should be doing next. And his presentation was breathtaking in the forward-looking nature of the statements that he was being made, as well as exemplifying his vision with videos and visual support of fully automated laboratory for genetic medicine. A genetic factory is, I think, how he called it in that presentation. And after seeing it, that's all I could think about and wanted to do: join Council, help him build this laboratory, make it a reality. And my entire career has been at this intersection of building and operating high-efficiency laboratory to provide capabilities and, well, let's call it value to patients that would not be achievable without living at the bleeding edge of the integration of technology in the laboratory to provide products and services. So the emergence of liquid biopsies, for example, is, I think, a perfect example of this, but also non-invasive prenatal testing, early cancer detection, et cetera, all of those life-changing products are possible because of the laboratories that are being built behind it, and have the opportunity to build a few of them and develop the expertise in that space. Find it very exciting to have a connection of everything that I do to patients, but also to the physical implementation of a factory, right? High throughput system. So yeah, that's been the thread for all those companies that connect them together.
You know, when you talk about human bioequivalence, how are you quantitatively benchmarking fidelity? Are you ranking it for trisonomic similarity, functional outputs, or longitudinal phenotype stability?
Yeah, that's a great question, right? So we, of course, have a great example of what we're trying to achieve. We are all one version of it, right? A human. So the measurement of the distance between what it is that we can do in the laboratory versus what we have set out to achieve is multifold, right? And there are layers that allow us to show how close or far we are from our target. the genomic profiling, right? And specifically, we think that transcriptomics is an extremely strong surrogate of a particular cell state and the function that arises from the cell state. And I will say it's not just transcriptomics, it's single cell transcriptomics, which gives us visibility into not an aggregate of cells, but a cell population with all the granularity of the cell types that form a particular cell-based structure, right? So if we think about trying to make a very specific cell type, say a pancreatic beta cell, then we would want to see a homogeneous cell population of pancreatic beta cell. But if we're trying to make a pancreatic beta islet, then we need to be able to see all of the various cell types in their distributions, right, that make up a human-like pancreatic beta islet, and that the ratio of all the cells is consistent with what we see in the human body, et cetera, et cetera. And then, of course, the function, right? So continuing on that thread for pancreatic beta islet, the insulin production under glucose treatment, right, is one of the standard assays to demonstrate whether or not the system perform as intended. And of course, we have very good baseline data on how a functional human pancreas should behave under a particular condition. And so striving to reproduce this at the functional level is helpful. From a platform and controls perspective, there are some constraints, the type of assays that we can use, right, to reach the particular state of interest. It needs to be fast, and it needs to be non-destructive to limiting the amount of material that we need to use or destroy to be able to take those measurements. But yeah, both functional and genomic is where we're at, and there are emerging modalities around the proteome that would be, of course, of great interest. We're not using this today outside of flow cytometry and surface markers, but we can do a lot more holistically with emerging technologies, and we are certainly aiming to move in that direction to get a full digital representation of a cell state that, of course, then needs to be mapped to function.
Now, this is great. I love it, actually. So I'm gonna go down through and bring it up. I got a lot of questions, by the way. I've read a lot of your research, just FYI, so I can dive into things pretty deeply. But I'm gonna raise it up a little bit. I have another show called Longevity, and what you're doing becomes part of what I'm building, right? And I want to talk to you about that after the show, because I think there's actually, I got one more question that I wanted to go down through. Is when you're going down through and looking at this, how do you address a gap between structural similarity and organoids and true functional equivalence, especially in complex systems like immune response metabolism? How do you where does that gap? How do you address that gap?
Okay, yeah, we're going real quick. So the structure leading to function, the example that I like to use for this is kidney and the crypts, right? And if you look at our advancements as a field, well, those type of systems are actually less advanced than the other ones, right? So if you look at, again, going back to pancreatic beta islet, liver islet, et cetera, and the relationship between the structure and the function is weaker than in the context of kidney and some other model system. And when we need to ensure that the self-assembly process leads to a very specific physical structure, we are starting to talk about the boundary and the bleeding edge of the field. And as a corporation, as a company, we do not want to be at the bleeding edge. We do not want to be at a level of risk that is scientific. And so we have not focused on this. Going back to the markers that we are looking at, right? If we want to be able to control structure, we need to be able to measure structure. And that leads us to imaging. And while we are doing imaging as part of our standard assays, as everybody else is, being able to map this back to a control algorithm that allows us to ensure the emergence of the exact same physical features that we are looking for is beyond the scope of our capability today. As a matter of
How
fact, it's
far is
part of
that
our.
out? And what do you think, right? Because that's where we want to go, right? But how far do you think this is out? And um, actually, why
Um,
don't we do this? I want to get you on my longevity show because we can dive in deeper to this stuff. I have a lot of MDs and double PhD, the folks you have at Carnegie. I want to dive into it a bit more. Let me do this. Let me take it back up a little bit. I want to say, what was the aha moment that led you to focus on bioequivalence at scale? And and then the second part of the question, what has changed technologically that makes it possible today versus 10 years ago, right? It's different today.
yeah, two great questions. So I think the aha moment is actually my work at Chemify, right? Showing that we can, the parallel with chemistry is, I think, relatively strong. Right? In chemistry, we start with, okay, this molecule based on my AI modeling should cure that particular disease or should bind very well to that particular pocket in the protein, et cetera, et cetera. Right? So I start from the end, and then I figure out everything that is needed to get there. Right. And there's also a really interesting consideration on IP, right? As I go to that new composition of matter, I cannot find the same space. I cannot reuse some of the protected reactions that are out there, et cetera, et cetera. So I need to find new space to get there. And when it was time to come back to the States and ask what's next, the parallel to what we want to do in biology is perfect, right? If we want to restore a particular degraded tissue, and we have an example of what the healthy functional one is, and then we have the technologies, or sorry, the ambition to do this in the laboratory, and then it's about figuring out which path allow us to get there. And if we continue on this parallel, right, we need the ability to we can start to think of and use cells and their plasticity, which has been the
Mhm.
primary challenge in the space to I would say being able to create high quality, reproducible, scalable manufacturing processes. The fact that cells are alive, they're changing all the time, they're talking to each other, they require a mutation in the laboratory. Cells at passages 1, 2, 3, 4 are not the same cells anymore, right? The fact that the systems are alive and changing all the time means that we need this type of active control system
Yeah,
to
yeah.
get them to be. So it's a high throughput synthetic manufacture, low latency, extremely low latency synthetic chemistry system. So the aha moment is, okay, what I've done there is directly applicable,
You
most
said,
likely.
I missed what you said. Did you say ion? Is that what you were saying? I missed there was a word I missed.
Sorry, no, I didn't say ion.
You
I'm
said
not
something
sure.
about immune. I couldn't understand what you said.
Uh,
Immune, maybe. There was like two words I missed.
I said low latency control system. Is that
No,
maybe the yeah.
it was something
Okay,
else, but
no.
I'll do your research and check it out. It's very interesting. Um,
Okay.
you have a very unique background, right? And if I recall checking through your LinkedIn profile, you have a background in applied physics and biomechanical engineering. That significantly has to change the way that you approach biology, right? And um, does that do you look at things differently because of your background? And has that helped you build companies?
An interesting question. Yes, I think it forced me to, for one simple reason. I'm not a biologist, and my long-term memory may not be as good as a lot of other people. So I struggle to go into the details and remember all the names of the molecule and blah, blah. So it forces me to think in terms of primitives, right? Of the key logical components that are relevant to biology. And that's super helpful to then build engineered systems that are systematically addressing subcomponents of,
Oh, I like that. I like that. Interesting.
and so I have to simplify everything that I hear in a way that I can both remember and build upon. And I think this is maybe a key characteristic necessary to design and build platforms, right? Where we need to start
To
from.
design and build everything. Sometimes we forget, right? That it's like building a house. It's a foundation. I keep it simple, right? It's like building a house. If you have the foundation and it's a little off, nothing good happens to the rest of the house. And the same thing, I like that approach. How do you? Let's talk about this for the audience out there. We talk about human bioequivalence. Can you say just in practical terms, for the audience, what does that mean? What is bioequivalence?
Yeah, that's the term that we find in scientific literature here and there. My own definition is exactly the same system that we have in our body, right? So taking a,
Exactly. Digital
you know,
twin, right?
um, yes, I think so, right? With a level of, to your, I think a couple of questions ago around how do we measure the distance between what we have in our body and what we have in the lab. What we have in the body, the standard is, making that becomes bioequivalent in the laboratory, and being able to, demonstrate that indeed we have recreated the exact same cell that we have in our body. Yeah.
Okay, so we have traditional cell structure and we have organoids and we have animal models. How are those different and why are they better? Why are organoids, how are they different from traditional cell structures or animal models, and why are they better, in your opinion?
Yeah, great question again. So I will remember similarly that that is a presentation that happened in my PhD that I still remember relatively clearly. And the statement was made by an FDA scientist, and it went as human and not large mice, right? It was a big slide with a mice there that was crossed out, human and not large mice. So using animal models or surrogate for human biology is limiting as a matter of fact, right? They're sure we're mammals, but a completely different animal system. The correlation between the response to a particular drug of a mice versus a human is is very different, right? And I don't remember exactly the number. I read a study or a few studies on that topic, but I think the correlation is something like 27%, right? Where the rest of it is either non-correlated or anti-correlated, which means using a mice to try to understand human biology is inherently limited. And here's the thing from an engineering perspective, it's never going to get better. The mice is going to remain a mice, and we, actually, that may be not fully true in the sense that we can humanize them in some ways and improve them in some way, but the core is just fundamentally different, and there is just a limited thing that we can do. The opportunity with cells, organoids, and then in vitro tissues and some of the other techniques that we use is, of course, to use the real biology that we are trying to work on, right, to either cure or improve or understand, right, that
Because
is
we want to make
fully
sure the
well.
efficacy is right. We got to make sure the efficacy of what we're doing is right. And sometimes we've made assumptions based on models that weren't 100%, right? Because if you got mice models 20 to 90%, depending on what you're doing, well, maybe it's 20 or maybe it's 90, right? What if it's off? Nothing good
Sure.
happens. But I mean, and so when we talk about the future, Daniel, what you're doing is building models that replicate the human condition, the human cell, so that we can make sure that whatever we're doing is much more effective, right? And we struggled historically to model human biology, correct? Right? I mean, that's what
Correct,
we're
yeah.
going. Do you
100%.
think?
Otherwise, we don't need animal models for sure.
And are you talking about incremental improvement or complete paradigm shift in how medicine is built? What do you think in terms of the model? It changes things, right?
Yeah, and that's a fantastic question. And I think the answer to that, unfortunately, is both, right? That we are proposing a new paradigm that will allow us to have eventually true bioequivalent cell organoids and tissues of what we have in the body. However, this is still going to take years, many years, but we are already at the point where we can make substantial impact today on drug discovery by improving on the status quo that is mediocre. There is no better measure to how little we control in modeling human biology or understanding modeling human biology than the rate of failure of drug discovery, right? It is one of the most valuable endeavor, both ethically and commercially, to develop new drugs, and yet the clinical success rate or the drug discovery success rate is, I think, 9%. Right? So 91% of the time we get it wrong. Can we do better than this? Yes. FDA's mandate to remove animal models is a momentous event in our ability to work on that number. We have been inherently limited by having to run our drugs through mice and dogs and other animal models that are just not representative of human biology, which means we are eliminating compounds that would work, or drugs in general, right? They don't just have to be compounds; it can be biologics, et cetera. So we are removing compounds for the pipeline that don't work in animal but will work in human. And similarly, we're selecting compounds that do work in animal but don't work in human. And so being able to rebuild the drug discovery, drug development, and drug selection pipeline around human models of increasing quality, right? As we improve our technology, as we iterate on those models, we will iteratively improve the rate of success of those drug discovery. And I think our approach brings something to the table that is really interesting from an engineering perspective, right? Now that we can define and design the type of model that we're looking for with data, which also means features, right? We can ask this question: why did this compound fail? And let me improve my models so that as a standard, this failure never happens again, right? And improving our models and model set so that over time, a failure never reproduces, and we inch that rate of success of drug discovery higher and higher by having better model for drug discovery and drug selection. One parallel that I like to make around this capability, right, is the equivalent of the annual release of a Toyota Corolla, right? New features, new capabilities, et cetera, but for human models of drug discovery, right? So my liver model didn't catch that particular toxicity. Why? Let me improve my liver model so that it catches it, and little by little catches all of the critical features of biology that the liver performs in the human body for drug discovery, and get us to a much more efficient paradigm for drug discovery. And by the time we have reached bioequivalency, we've of course changed everything, but the path there is value creative immediately. And so, of course, from the perspective of building a business,
there's no choice. They're inefficient models. So here's the thing, right? You talk about discovery, but really, using the AI that you're using now, there's other leverage, right? And where does it create the biggest leverage in design, prediction, automation, or discovery?
Sorry, that broke up a little bit. I think the other leverages of impact
So
and drug.
you're combining AI, robotics, and biology, right? But where does the
Yeah.
AI create the biggest leverage? Is it for the design, prediction, automation, or discovery? And you're going down through this process. Where does it help you the most?
That's a great question. So I think AI is the enabler of our entire approach, right? Automation existed a long time ago. Process control existed a long time ago, laboratory efficiency, process, et cetera, et cetera. So the inflection point to be able to control the cell manufacturing process and iteratively get towards bioequivalency, AI is a step function. Everything else is the price to play, if you will, right? Like without automation, we cannot realize all of the necessary impact of AI, right? AI is in a large corporation, a very small part of the organization, right? It's just an algorithm that then needs to flow out all the way to the laboratory and get executed on, et cetera, et cetera. So we need every single one of those components that I've just mentioned to be, exceptional, right? One interesting element, right, is I try to buy everything that I can so I don't have to reinvent the wheel. But for cell state control in 2026, we know most, if not all, of the variables that impact cell state, if you will, right? And that would be, of course, the media composition. That would be, the cytokines, the growth factor, but also the temperature, the pressure, the relative gas concentration, the surface chemistry that the cells are exposed to, and a single system that controls all those variables does not exist. And so we had to of it, and we had to make sure that our AI can talk directly to it through APIs, et cetera, et cetera. And so that is part of the solution that we have to provide. So at the end of the day, we have true mastery over the manufacturing process for anyone's cells and any cell type, organoid type, or tissue type, right? Going back to platforms, right? At the end of the day, under the hood, it's a very simple computational primitive.
Mm-hmm.
State one, what is the current state of my cells? State two, what is the target, right? Or is the bioequivalent state that I'm trying to reach? AI compute the transformation function to go from state one to state two and apply in the incubators, right, in the real world, in the factory, the condition that you have predicted to get the cells to where we want them to be. Another element of this, right, is that at the end of the day, right, that the long-term objective.
This is going to change everything, though, Daniel. This is going to change everything, right? When we we go down through with these models and make them more accurate and replicate the human condition, we're moving towards the digital twins powered by organoids, correct? I mean, that's where we're going with this. And that's going to fundamentally change everything. And that's going to add, everything becomes more effective, right? And more efficient once we have those kind of models, correct?
Absolutely, yeah. All of drug discovery becomes more efficient, but there is also the exact same solution that we need for cell therapies, right? Like the same technology solution, the same achievement of having reached bioequivalency is what would allow us to restore function systematically to the human body.
So when we look at it, we're going to dramatically accelerate experimentation because we're using autonomous or agentic AI systems to be able to help us, right? What kind of orders of magnitude do you think, just in your opinion, is it going to change? Just so the audience sees a picture, right?
That's a fantastic question. I think it gets back to the paradigm and time. On a long enough timeline, we will not need the laboratory anymore. Right, we will have built a predictive data set that is large enough to make accurate in silico prediction that are working every single time. Of course, today we're pretty far from this, right? In vivo data is not large enough to get perfect prediction. A temporary phase where we need those surrogates, right? Cells, organoids, in vitro tissues, right? That would be more advanced version of organoids or more complex version of organoids, to acquire those very large data set that are perfectly recapitulative of human biology to then power this next layer of prediction, right? And the vision, of course, here is personalized drug for any ailment that exists. Does not break any law of physics, so it will happen. It's just a question of time. And the limiting factor is the quantity of high quality data to power those algorithm. Is going to take a little bit of time, but the line of sight to we don't need laboratories anymore exists. Don't have a crystal ball to tell you when. It's probably in the decade-ish timeline that we'll need to get there. But yeah, so at some point we purely in silico prediction will be accurate enough that we will not need the laboratory part of those processes.
I mean, you talk about, now I understand your approach, right? And we're really starting to scale biology like we've scaled software manufacturing, correct? I mean, you look at it in a different, that paradigm. But at the same time, Daniel, we have bottlenecks, right? Cost, reproducibility, regulation, or is there something else that I'm missing?
An interesting question. I mean, I think you've highlighted, at the end of the day, everything is cost, right? Like if there were no cost denominator to what we're doing, we would be able to do anything.
I mean, cost of time, right? There's cost of time. Do we have the time? How much time is it going to take? And can we do it in a reasonable amount of time? Absolutely.
Yeah. So similarly, right? Time is cost and money, but the key limiters to, well, some of the possible futures, right, to some extent is know-how and technology and the ability to get there, right? We've talked about the frontier of model development, if you will, being being able to control the structure of the system that we were building. But human biology is, of course, so many variables, right? The order of magnitude of variable that need to be controlled is extremely high, and so to some extent also depends on what future we're talking about, right? On a long enough timeline of what those technology would allow us to do is also to improve the human condition, right?
Yeah.
The
Well,
one way.
you've got to ensure the consistency and quality when working with these living systems, right? And it's a difficult. It's not. I mean, it's like a whim de jour with some of the biology out there. So how do you ensure the consistency and quality, and then continuously sense so you're monitoring it if it does change?
Yeah, so I mean, we've established the absolute measure of distance between what we're trying to do and what we have done earlier, and then it becomes a problem of convergence, right? So having those control algorithm accurate enough that their output, consistently and reliably shortens the distance between where we are and what we're going to do, and then the acceptability criteria being how far can we be from the target to accept a new
Mhm.
model, which is something that over time should become smaller and smaller, right? And so you're absolutely right that those are or limiting factors. I think reproducibility today for increased complexity system, even organoids, right? I'm not talking about structured tissue just yet, is something that gives users pause, right? If I order one batch of organoids in March and I get a particular set of results, and I run that experiment again in June with a different set and get completely different answers, there is, of course, no confidence possible into the reliability of or the trustworthiness of those results, which to some extent means the system cannot be trusted, right? And so,
Right.
first,
That's the and if
see
that happens, nothing good happens, right? When we start not trusting it.
That's right. And if we think about organoids, right, it's the combinatorial variance of all the underlying cell type that self-assemble into a complex model. So it's the combinatorial explosion of the sources of variance that yield, in the organoid batches, high variance from batch to batch. And so from an engineering perspective, right, we need to master each cell's control algorithm to be able to control the assembly, to then be able to, the set, if you will, to then control the assembly. So we have, they are combinatorial, right? So we cannot exclude one from the other in terms of the control system that we need to put in place, but demonstrating that we can solve the simpler problem, right? Before going into the more complex one and then the more complex one. And for example, at the homogeneous cell population level, going back to the example of making a pancreatic beta cell, one of the key challenges in the industry is autologous cell manufacturing. That means, Gary, if I take your cells and if I take my cells, the differentiation protocol that we'll have to apply, we know is different, right? Because our biology is different enough that trying to ignore this and apply the same fixed protocol doesn't work and yield a lot of variability. So one way to demonstrate that those systems work is being able to computationally predict the adaptive nature of the protocols that will work with a particular biological state, is one such demonstration of capability. And once we have demonstrated this, then let us go to more and more complex systems so that we can crack reproducibility at a level that we can achieve instead of taking too big of a step and failing.
It sounds to me like you're building a biofactory for human cells, right? It sounds like that's where the market's going. And when we look at that, what do you consider yourself? With your own background, are you a data center, a lab, or a factory? What do you think we're becoming, and your infrastructure? Because it sounds like all
That's
of the above.
So currently we're a lab, right? And
Really,
we're
are you a lab or a data center? Because it sounds to me like data. I got to tell you, I know what you're saying, but I have a little bit of a background in AI myself. It sounds to me like you're a data center.
I mean, there's a heavy component of data center, of course, but that's only half of what we have today. There's a laboratory that feeds the data into the data center. Then there's the prediction from the data center that don't pop up in your UI that actually hits the lab
And
to make it
that becomes a factory, Daniel, right? So we move
that
into a
eventually.
So if we look at it, so that's a nice picture. Just for instance, when you're going down through and talking about what you do, if you want to, I talk about storytelling, painting pictures with words. All of what you're doing goes into what longevity, right? And I'm right a lot about. I've written like 900 articles and longevity because what you're doing is helping us to be able to live longer and increasing the quality of our lives, right? It's a wonderful picture to paint. I love it actually. Um, so, based on your own background, how important is automation and robotics in achieving true scale of going down through building your data center lab factory?
Yeah, so notwithstanding software automation, right, which is a given, we cannot achieve scale without automation. And it's a question, of course, of cost, right? Being able to displace labor to make things affordable is a critical side effect of automation. And for multi-month, multi-quarter manufacturing processes to make highly mature cell type, we can talk about the transformation from completely unaffordable to, well, the labor cost is negligible in the big picture. In biomanufacturing, we have another limitation, which is talent, right? Trained individual that I can actually run an end-to-end campaign and have enough understanding of the biology and experience in running a particular differentiation to be able to process the data, right, and make informed decisions based on that data. Very scarce, very rare, very difficult to hire, and inherently a limiting factor to the scaling of the system. And one way to think about automation being not just the robotics, but
Do
the
you
so
do you
again.
hire somebody that's got a PhD in data science? Because you're talking, I'm thinking about how much that person's going to cost too. By the way, it could be a lot of money.
So
Because what
of
background would
course.
that person have? By the way, we have a large audience that watches GSD. What kind of background would that person have?
So I mean, that person would typically have a PhD in iPSC differentiation sciences, probably a postdoc, and have spent many years, right, 5, 10 years to a decade on a very specific tissue type, right? And if the ambition is, well, we need to make liver, pancreas, heart, neuronal models, et cetera, then we need all that breadth of expertise. And today, all of those positions need to be filled to plain and simply being able to manufacture. And once you have a PhD and spend 10 years developing a skill set, learning how to advance science and what have you, manufacturing is not the most appealing job possible, right? It's like a pretty repetitive, task-driven work environment, and yet that level of training is what's needed to deal with human cells. One way to think about AI in industry is the download of human know-how into a centralized and scalable system. And so a lot of what we do is work with, my co-founders are, of course, great example thereof, the very best scientists on a particular topic and encode that knowledge, right? And so the database and the AI decision system are a version of the human decision-making system, except once it's encoded and once it's accessible, it is immediately available in North Carolina, in New York, in California, in Texas, across the world to make consistent decision at any site, anywhere. So a key element of the automation and the scalability, and as we mentioned, reproducibility, right, which is just not batch to batch, it's also site to site and operator to operator, is that encoding of knowledge and the consistent decision that can be made from that encoding for all differentiation campaigns. So that part of automation is critical. As you know very well, the implementation of AI is also not binary, right? It starts with enhanced human decision, then it ends up becoming human in the loop for verification, and then it's truly autonomous. But every step on that axis makes the human more efficient until, well, it's how to say, superseded, if you will, by just the quantity of knowledge accessible to AI that will never be parsable, if you will, by the human brain. So yeah, and then of course the physical automation thereof is a critical element of reproducibility and scalability, right? To be able to control very large bats, we need automation, we need a automated system.
We're going to see some breakthroughs in diseases. Uh, and, one of the things that came up based upon what you're telling me and my own background is we're going to have incredible regenerative therapies at scale very soon, right? It's not going to be that far in the distance. Um, and the profound impact on humanity, right? Those regenerative therapies, that's myself, right? And being able to turn the clock back. How far do you think we are, and what diseases or areas do you think we're going to find major breakthroughs first? So, first question: what diseases or areas, and then secondly, what about regenerative therapies?
Yeah, so where are we? I think we're right there, right? We are at the cusp, and we're at the beginning of the adoption of this technology, right? What you and I were discussing before this call. Clearly, we're there, right? And the question is the breadth of adoption and the accessibility of those technologies. So, we are in this. That's a big driver of us at the company on working on this problem. We have clinically validated cell therapies for a wide variety of, let's call them diseases, right? So Parkinson's, for type 1 diabetes, et cetera, et cetera. We have clinical trial data that shows those therapies today, a few years ago, actually work, and it's about the late stage regulatory, but critically the manufacturing challenges for those therapies. And as you asked very early on, what have I been doing with my career? I've been building a laboratory solution that allows us to crack those highly complex problems. When we think that today, with those adaptive biomanufacturing, AI-driven system, we can actually get to the point where we can make those things affordable, that we need to decrease the cost 2 orders of magnitude, right? Like to go from a few $1,000,000 per dose to tens of thousands per dose, right? To see broad adoption. But we know that works. We know how to develop them. We know how to manufacture them, but at high cost. And we are at the Model T moment, right, of the Ford manufacturing pipeline, but for human cells, because they're alive, because they're changing all the time. We need to rethink the entire infrastructure from top to bottom, or bottom to top. And one of the things that we're bringing to the table that automation on its own will never be able to do is that adaptive nature to human biology, to cell biology, right? And so that will allow us to crack it. So where do we see this first? All over the board, all across the board, all at the same time is what I want to see, because it's the exact same problem that is limited, the cures for macular degeneration, the cure for Parkinson, the cure for type 1 diabetes.
I've personally seen, Daniel, when I had my mesenchymal stem cells done, right? I've had about 150 million put in me now over 3 treatments. I've seen people with Parkinson's, personally, Alzheimer's, dementia, supranuclear cerebral palsy. I've seen profound changes in those folks by using these mesenchymal stem cells for umbilical cord. 3 hours after a woman delivers, they've been cultured and they're put in IV into the body. I've seen profound changes. I mean, they're like miracles, right? And as close to the religious experiences you could get with biology. And you're on the cusp of it. Listen, I spent a bit more time today, but I love the topic that you have. And if you want to continue the conversation after we done with the show, that would be great. I love what you're doing, because for you, all of you out there, this is about, nobody wants to die. And what Daniel's building is something that's going to be able to prolong our life, give us a better quality of life, reduce the animal testing that's been done, and to make profound changes in terms of efficacy and efficiency, so that we all can live longer, happier lives. And I just, applaud you on what you're doing. It's great. And, this is part of the integrated approach that I talk about in terms of longevity, because, Aristotle said the sum of the parts of this whole, and so the Germans Gestalt. And that's what this is all about. This integrated approach, tying these pieces together, is going to change everything. And we talk about living to 150. This is one of the ways we get there. So I want to thank you for being on my show today. Closing thoughts Daniel, and how do people get a hold of you?
Thanks for having me. That was great. I think the question that you've asked and the mastery of the subject made for a really interesting set of questions, and some that I haven't thought of asking myself. How to get rid of me? So we have a website, IORGAN.bio, and my personal email is daniel@iorgan.bio, and we'd love to continue the conversation with you and anyone else who wants to chat about these topics with me.
That's great. Thank you for taking time out of your busy schedule to join us. And thanks to all of you. We're now approaching 1,700 podcasts that I've done. So check it out. And my new book came out, How Stem Cells Change Me, and it's on Amazon. Check it out. If you like it, like and subscribe. To all of you out there, stay happy, stay safe, and stay healthy. And I'll be back to you again soon with another exciting edition of GSD Presents Silicon Valley AI and Tech. My name is Gary Fowler, and I'm your host. Thank you, everybody, and thank you, Daniel.
Thank you, Gary.